इससे पहले जो मैंने अभी वीडियो आप लोगों के सामने भेजी थी दैट वाज रिलेटेड विद द रेस्पिरेशन, और उसमें जो रेस्पिरेशन के डिफरेंट इवेंट्स हैं उनका कोरिलेशन हमने बतलाया था कि किस तरीके से एक स्टेप दूसरे स्टेप से कोरिलेटेड है। तो जो अभी तक हमने डिस्कस किया था रेस्पिरेशन में, हम उससे आगे भी बढ़ेंगे, थोड़ा सा उसको एक बार फिर से रिवाइज करते हैं। तो बतलाया था कि रेस्पिरेशन के अंदर जो मेन इवेंट्स हैं क्या क्या हैं। द पार्ट 1 हियर इट इज द glycolysis. This all I am talking about is the aerobic respiration now. Okay, so first part is the glycolysis, and with this glycolysis is followed by oxidative decarboxylation, and the oxidative decarboxylation is followed by Krebs cycle. ठीक है, और उसके बाद में इंपॉर्टेंट स्टेप आता है दैट इज RETS, रेस्पिरेटरी इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम, एंड फाइनली देयर इज द टर्मिनल ऑक्सीडेशन। So what happens in these 3 steps, glycolysis, Krebs cycle and oxidative decarboxylation, obviously what happens, we obtain NADH2, we obtain molecules of the NADH2 and FADH2 also. In fact, these are in the form of NADHN plus HN plus 2 electrons. So these electrons, they move, they enter into the next step, there is the RETS. Now in detail we will study the RETS, or the oxidative phosphorylation. Oxidative phosphorylation in which the ATP gets synthesized. So it means today's main topic will be related with the RETS, that is respiratory electron transport system or transport chain system or chain. And during this process, because ATP is synthesized, therefore it is also called as oxidative phosphorylation. Oxidative phosphorylation. Okay. What happens in this event is very interesting. What happens that when there are hydrogen molecules, that is HIN plus 2 electron, they move, or they just move towards the, say, mitochondrial membrane, and ultimately they meet with half molecule of the oxygen to give rise to what molecule. Now these hydrogen ions, these have a very low RP value. However, this oxygen, it has very high RP value, that is redox potential value. This has been described in photophosphorylation. What is the RP value and how does it work? Low RP value, it means it has high electron energy and it has low electron energy. So it means when electrons move from this place to this place, from high energy to low energy, then energy is synthesized in the form of ATP. That is what is happening in respiratory electron transport system. It means there is downhill movement of energy. It is called as downhill movement of energy during respiration. Okay. So this RETS chain, its site is, if we discuss about the site as described earlier, here it is in the inner mitochondrial membrane. Inner mitochondrial membrane. In fact, enzymes responsible for this electron transport chain of the respiration, those various enzymes are formed in the forms of in inner mitochondrial membrane in form of certain complexes. So, for example, this is mitochondrial outer membrane, this is the inner membrane, and inner membrane is always in the form of certain invaginations called as cristae. So this inner membrane, as you know well, it consists of certain small particle-like structure called as the F1 particles. So besides these F1 particles, there are certain enzyme complexes which are found in this. And when electron moves across these complexes, ATP gets generated. Okay, so how many of these are complexes? What are their name? What are their components? Let us discuss it in detail here about the complexes of respiratory electron transport chain, that is the complexes which are found in inner mitochondrial membrane. Okay, there are a total of 4 main plus 1 more, 5 complexes, and these complexes are named as just like this. Number 1 here, it is the complex 1, written as the Roman 1. It is NADH dehydrogenase reductase complex. Complex 1 or NADH dehydrogenase reductase complex. Then there is complex 2. It is succinate coenzyme Q reductase. Succinate coenzyme Q reductase or complex 2. Then third is the complex 3rd. It is cytochrome BC1 complex. Cytochrome BC1 complex. And fourth important is the complex 4th, which is named as the cytochrome oxidase. Cytochrome oxidase. Besides these 4, there is one more, as I just told you, there one more complex is found. It is the complex 0, and here it is nothing else but it is the F1 particle, which is found in the inner mitochondrial membrane. Okay, so and besides this, okay, there are certain mobile carriers also. There are certain mobile electron carriers also. For example, coenzyme Q. So this is also found in between various complexes in the inner mitochondrial membrane. Okay, so what is the location of these complexes? Let us see here. I will write down. I will make these complexes in the form of certain boxes, and it is number 1. It is complex number 1st. Then it is complex number 3rd. It is complex number 4th. Then where is the second one? Second one must be in between 1st and 3rd. It is 1st, 3rd, 4th, and the position of 2nd is, I am just find it here, just above in between these two, 1st, 2nd, 3rd, and 4th. And here I am just drawing the complex 5th, that is the complex O or the F1 particle. Okay, so this is the position of various complexes in the inner mitochondrial membrane, right? So this is the position of various complexes. This is complex 1st, complex 2nd, complex 3rd, complex 4th, and complex O, complex 0. Okay. So we start here with. So here we start where it is the very first complex, that is NADH dehydrogenase. Whenever hydrogen or the electrons, they move from NADH2, then they enter into the complex 1. Okay, and when the hydrogen enters from FADH2, then these enter into the complex 2nd. So I just told you that during the complete period of the glycolysis, the decarboxylation and Krebs cycle, at several places or most of the places, the hydrogen is released in the form of NADH2, and only at one point when succinic acid changes into the fumaric acid, then hydrogen is accepted by FAD. Okay, so only in one case hydrogen is obtained by the FAD, and then that FAD enters into the electron transport chain in complex second. However, when it is in the form of NADH2 at other places, it enters into the complex first. Right. So here it is the NADH2 which has been formed at various places during the Krebs cycle, glycolysis and oxidative decarboxylation. This NADH2 releases hydrogen, that is in the form of ions, as well as the electrons. Those hydrogens are accepted by FMN, flavononucleotide. This FMN, it means, gets reduced to FMNH2. Okay. Then soon it gets converted back into the FMN and it donates hydrogen. Those are accepted by iron sulfur protein. It gets reduced. Okay. Again, it will get oxidized. It will release hydrogen. Those hydrogens are accepted by coenzyme Q, which is the intermediate mobile carrier in between first and third. Okay. And then again, this coenzyme Q releases hydrogen. It gets oxidized. Then those hydrogens are accepted by cytochrome B, like this. It means this is the absolutely oxidating reduction process always continue depending upon the RP value of various substances in between various complexes. तो फिर से यहाँ पर NADH2 से आया हाइड्रोजन आया, किसने एक्सेप्ट किया? FMN मोनोन्यूक्लियोटाइड. ओके, तो ये रिड्यूस हो गया, वापस ही अपने हाइड्रोजन छोड़ेगा. ठीक है, those will be accepted by the iron sulfur protein. It will get reduced. इस तरीके से continuously ये एक्सेप्ट करते हैं, रिलीज कर डोनेट करते हैं, एक्सेप्ट करते हैं, डोनेट करते हैं, एक्सेप्ट करते हैं, डोनेट करते रहते हैं. Like this, the process continues. Here again iron sulfur protein, like this, again like this, again like this, again like this. Okay, so what happens when electrons move across this NADH dehydrogenase? At that time, here ADP changes into ATP. ADP changes into ATP in the complex 1. Right. Similarly, when electrons jump, they just move across the complex third, there also ADP changes into ATP. Okay. And the process continues. Again, this is accepted by the intermediate cytochrome C here, and it will get again oxidized, reduce, like this. Okay. Cytochrome A and A3 are always formed together. Again like this, continuously reduction oxidation process goes, ultimately like this, and hydrogen ion, those are say available just after the cytochrome oxidase complex 4th. And here also at this step the ADP changes into ATP. Right. And this mechanism of the conversion of ADP into ATP, basically it occurs in this F1 particle. Here it means it is the F1 particle where ADP changes into ATP. Now let us discuss about this one. When succinate changes into fumarate in the Krebs cycle, at that time hydrogen are accepted by FADH2. अब ये जो FADH2 है, ये enter करेगा कहाँ? Not in the complex 1, but it is accepted by complex 2nd. And again the same process continues here. ये यहाँ होगा, ये हाइड्रोजन एक्सेप्ट करेगा, डोनेट करेगा, रिड्यूस होगा, और यहाँ से ये हाइड्रोजन कहाँ आएंगे? इंटरमीडिएट मोबाइल कैरियर में, that is the coenzyme Q. It means when FADH2 releases hydrogen, then those are accepted by the complex 2nd, and then from the complex 2nd this goes to the coenzyme Q and finally to the complex 3rd. As you see that here it is formation of one molecule of the ATP, here also formation of one molecule of ATP, and also formation of one molecule of ATP. But what is with the complex 2nd? No. none of the ATP molecule gets synthesized in complex second. इसका मतलब यहाँ पर ATP का सिंथेसिस नहीं हो रहा है. अब ध्यान देने वाली बात यहाँ पर क्या है? It means कि whenever NADH2 enters into the electron transport chain, it releases 3 molecules of the ATP. However, when FADH2 enters into the respiratory electron transport chain, then it releases 2 molecules of ATP. This is one major difference between these two. Now what happens here? Now here it is the point where these hydrogen, those are accepted with half oxygen to synthesize water molecule. And this is the step, last step, that is the terminal oxidation. So to continue the chain, for the continuous production of the ATP, it is must. If you don't have oxygen at the end, it means the chain cannot be continued, it cannot be completed, ATP synthesis will not be there, and ultimately the organism cannot respire, no synthesis of the energy. Okay, so in the just coming diagram, what I shall be discussing is how this phosphorylation, how this ATP AD is synthesized in F1 particles. तो ये तीनों कॉम्प्लेक्स से जब इलेक्ट्रॉन ट्रांसफर होते हैं, तीनों कॉम्प्लेक्स से जाने के बाद हर एक कॉम्प्लेक्स में एक एक एटीपी का सिंथेसिस हो जाता है. So it means this is very key factor for the production of ATP in all these complexes, right? So what happens during the phosphorylation, or how the ATP is synthesized? Let us see here in the F1 particle. One very famous theory, chemi-osmotic theory, was proposed by Mitchell in 1961, who has just discussed that how the ATP can be synthesized in F1 particles. Okay, so here it is inner mitochondrial membrane. Okay, it means here it is the mitochondrial matrix where F1 particle is found, and here it is the intermembranous space. चूंकि बाहर की तरफ आउटर मेम्ब्रेन, ये इनर मेम्ब्रेन माइटोकॉन्ड्रिया की, इनके बीच का इनर मेम्ब्रेनस स्पेस, इंटर मेम्ब्रेनस स्पेस, ये हमारी हो गई इनर माइटोकॉन्ड्रियल मेम्ब्रेन, ये माइटोकॉन्ड्रियल मैट्रिक्स. सो जब इलेक्ट्रॉन ट्रांसपोर्ट चेन हुआ तो क्या होता है? हाइड्रोजन आयन कंसंट्रेशन अब यहां जो हमारा इंटर मेम्ब्रेनस स्पेस है उसके अंदर बढ़ जाती है. देयर इज हाई हाइड्रोजन आयन concentration, and here it is the low hydrogen concentration. So once these are formed, then these have to go back to maintain the equilibrium. यहाँ आ गए, यहाँ पर आते, अब ये वापस जाएंगे कहाँ? माइटोकॉन्ड्रियल मैट्रिक्स में, but their entry is banned through this inner mitochondrial membrane. Then what happens that these hydrogen, they enters through this side. This is the position. ये इसका head है, ये इसका head है, ये tail है. तो hydrogen, they always enter into the mitochondrial matrix through this F1 particle. Okay. अब जैसे ही a proton gradient is formed, a proton gradient is formed, you have seen the osmotic potential theory. तो प्रोटॉन ग्रेडियंट की वजह से लो है, हाई है, तो उसके बाद में इसको मेंटेन इक्विलिब्रियम करने के लिए अगेन दे विल कम बैक. तो जब ये आगे दिस एंटर थ्रू दिस, और जैसे ही ये हेड पार्ट ऑफ द F1 particle is present, here there is the enzyme ATP synthetase. Just like this, when the proton move, then there is some shrinkage type of mechanism, and just it splashes the hydrogen like this. So hydrogen are moving just like a splash mechanism, and then as soon as the hydrogen enters, the ADP changes into ATP. So this is how ATP is synthesized in F1 particle at the last. So this is the most important event. How come the respiration? You have seen that the final aim here of the respiration is the synthesis of energy. That is ATP. So this is how the ATP is synthesized, and in just my previous diagram I had told you that how the terminal oxidation occurs, that the oxygen combines with the hydrogen to complete the chain. So this is all about a basic idea of the respiration, where I have tried to discuss at undergraduate level that how those things are interrelated with each other, how electron transport chain goes, how ATP is synthesized. Thank you.